So our next speaker is Thomas Hartung, who's a professor at Johns Hopkins Bloomberg School of Public Health. And he's going to speak about alternatives to animal testing and also a bit of AI. And actually, we talked yesterday because when you submitted your presentation, some EMA colleagues have concerns about the pictures and the figures in your presentation, whether they were copyrighted or not, because if we're going to publish this, and you told me last night, there's no copyright on any of your pictures because they are all AI generated.
Yeah, that's the modern times. So first of all, happy birthday. I had the privilege to work with EMA since 2002 when I joined the European Commission to become head of ECVAM, the European Center for the Validation of Alternative Methods, so exactly the topic I'm still working on. And EMA has been a trusted partner since, and I really appreciate to have the opportunity to be here. The topic is actually much more AI, not just as an afterthought, and if I had given the title myself, I would probably have said, AI will only replace regulators who don't use it. So, I made my slides available. The QR code is also on the last slide again. I'm directing since 16 and a half years now the Center for Alternatives to Animal Testing at Johns Hopkins University, so I changed the side of the pond. But I also co-direct the Center for Alternatives to Animal Testing in Europe at the University of Constance with Marcel Leist. And I'm also field chief editor of Frontiers in AI. My quest is to replace the dance around the golden rat and retire these type of animal models. When I joined the European Commission, I got the job because I had just successfully validated what is now known as monocyte activation tests and had developed one of these assays myself. These assays are now replacing in Europe alone 150,000 rabbits per year, but it took us almost 30 years to get there. Yeah. So this is showing you something. There's potential of modern technologies, but it is slow. And I want to show you that these things are accelerating because we have new technologies. Also, as a pharmacologist and toxicologist, did my share of animal experiments. I never liked them, but I did. When I was charged with replacing animal testing, this was mainly because of ethical concerns. Yeah? Find something which is the same or hopefully better, but mainly ethically. There's people who want this to be replaced. But the longer I'm in this business, the more I see this as an advance of science and an economical question. Yeah? These tests do no longer serve us well. This is an article I wrote last year, targeting a general audience about the misleading role of animal models in drug development, where I show that from a lot of studies combined, we have to assume no animal test is more than 80% accurate. Many of them are much, much less so if you compare the same experiment done on mice and rats or rats and rabbits. It is typically between 60 and 80%. And if you use such tools to find a drug to go into human trials, it is very often misleading. And this is alone should open up for other tools. The other thing is I never liked this war of faith, that there is some people who accept animal tests and defend them vigorously, and other people say we have to get rid of them tomorrow. So I thought early on in 2002 already, can't we do something similar to clinical medicine, where evidence-based medicine has helped us to be simply factful, demonstrate what is the fair place for each technology and compare them. So we created the evidence-based toxicology collaboration. In 2007, still at the Commission, we organized the first international conference and we got it started. Since 2009, I have a chair for evidence-based toxicology in Johns Hopkins. And the collaboration we created 2 years later enjoys strong support from agencies like the FDA, the EPA, but also our president is from the European Food Safety Authority. And I would very much like that EMA is joining the club in our board of trustees and others, because I think it is a timely thing to do to handle more objectively and transparently whatever facts we have to handle. So, invitation, number one. To just show you with one slide the difficulties and some of the problems, the chemophobia we have been produced with some of these animal tests, I give you the example of the cancer bioassay. The cancer bioassay is still the only way accepted to show whether a substance is producing cancer or not, and it is hardly done. Now, we have about 3,500 of probably 350,000 marketed chemicals which have ever done a cancer bioassay. And the reason is simply, it takes you 4 to 5 years to get the results. You pay about $1 million. You need 400 animals, and you need 10 kilograms of the substance to treat these animals for 2 years of their lifetime. Tell a chemist to produce 10 kilograms of a substance which is not yet in production. And the results you get are terrible. 53% positives. We estimate that maximum 5 to 10% of these substances are actually carcinogenic. The reproducibility between mice and rats is 57%, which is not really very much encouraging that they predict humans any better. And not even the organs in these animals are anyway correlated, the one in the kidney, the other one in the liver, and so on. So it is not really a value proposition to use this assay, and I'm so excited that ICH is finally tackling this to make this something of the history. In general, it is all about now transitioning to new technologies. This is an article from last month where I wrote about how this is similar to 110 years ago when horse coaches were being replaced by cars. And the type of arguments you hear, how reliable the horse is, how much infrastructure we are missing for the new technologies. And I very much hope that in a few decades, people will smile about these antiquated arguments and say, how could they ask about not using AI, not using modern technologies? So from my point of view, there's at the moment a number of disruptive technologies. They're disruptive because they're upending the way we are doing things so far. And I will focus only on 2 of them. One is the microphysiological systems, the organoid and organ-on-chip systems, mainly fueled from stem cell technologies, and then AI and data analysis technologies. Both of them feed into AI in making sense of these big data. So we have, on the one hand, the possibility to produce human relevant data on scale. And at this other time, we can analyze now data, because since 2 years, actually a machine is better capable of reading a scientific paper and annotating it than a human. But it's not limited, like my PhD student, to 1 or 2 papers per week. It is millions in a day. And these systems do not forget. And this is a tremendous value proposition. And this is allowing us, first of all, something which in the chemistry field we call green toxicology. In medicines we call this fail early, fail cheap, because we can use these technologies not after we spend almost $1 billion for preclinical development, and when we want to go into humans, when we do our 10 million package of safety testing, but we can do this even before we synthesize a substance if we have reliable computational methodologies, and we can do it on micrograms of quantities we have in our hands with the MPS type of system, the microphysiological systems, as the EPA is calling it. So these technologies are allowing us to front load already these concerns. The second side, which I think is really getting more and more interesting, and I will come back to both of them, is that we can combine this also with exposure sciences. Our mass spec systems are getting about 1 log order more sensitive every decade. We're finding more and more substances in the biospecimens, in the blood, in the urine, in biomonitoring studies. And again, it is the AI, the exposome intelligence, which is helping us to make sense of this and can lead us to human exposome or to personalized medicine, where we consider a treatment not as something for everyone, but something for an individual with a specific disease history and its unique aspects, which we more and more integrate by digital twins. That's the big, let's say, 10,000-foot perspective. Let's talk about how this vision for safer products, better treatments, and safer people can actually be implemented, and how these technologies are at this moment lending themselves already to do so. So the first technology, which developed really over the last 2 decades, is microphysiological systems. That's the umbrella term coined by the FDA for 3D cell culture systems, organoids, organs on chip systems, multi-organ on chip systems, patient on chips, and others. My group has been accompanying this over the years. We started with the first workshop report in 2014. We had several MPS workshops dedicated to the future, bringing opinion leaders worldwide together. The last one published just in April. We developed good cell culture practices for these systems, so to have quality assurance. And we started a series of world summits in 2022 in New Orleans, 23 in Berlin, 24 in Seattle, and just 2 weeks ago in Brussels with 1,500 people, a very successful meeting. And you can see here how the number of attendees has reached almost 1,500 now. The number of exhibitors is steadily increasing, a very vibrant activity which we had in Brussels, and we're looking forward to go to Washington in May next year. This is me handing over the baton to my wife, Lena Smirnova, as the president of the society who took over the organization of these things. And so my team was last time, responsible for the meeting. So our interest into organoid systems came from this steady increase in autism in the US. You see here the latest numbers from April this year. Now 1 in 31 children in the US is diagnosed before their 8th anniversary with autism. 1 in 31. This was 1 in 10,000 in the 70s and has tripled over the last 20 years, where no change in diagnosis also took place anymore. And this cannot be genetics, though there is a genetic component, there must be more of this. And it seems even to accelerate if you see the latest numbers, which might be possibly even a consequence of the pandemic. So we already 20 years ago started to work together, and as I say we, my predecessor in CAT, Alan Goldberg, and me at the time in ECVAM, and with 5 international conferences and 9 workshops, we addressed this problem, because there is only an animal test for developmental neurotoxicity, which takes you 2 years, but more than 1,000 animals, and thus cost 1.4 million to carry out on a single substance. Not really a value proposition to find and screen chemicals and drugs for their contribution. This is a paper from last year where we describe these 20 years, where really a group formed and ultimately led to OECD guidance on how to use in vitro systems for this purpose, because really a community out of nothing was shaped to address the right systems. And this was the meeting our European branch carried out last April in Constance. We developed out of this the interest into brain organoids from stem cells. We were not the first to produce them. We were beaten by half a year, but we were the first to mass produce brain organoids. So we really get about 4,000 human brain organoids in a 6-well plate. Yeah? That's about 1 cent of one of these. Yeah? And most experiments can be done on a single brain organoid. And these systems are not only having all types of neurons we have looked for so far, which are electrophysiologically active. We have oligodendrocytes myelinating the axons, a very rare thing, a very important endpoint. And we have astrocytes, and with the latest protocols, we get the glial cell population to about 50%, which is physiological. And all of them together, standardized. You see the size distribution. Here, the reproducibility in cell populations we must produce, and we can do this of different patient backgrounds. So, for example, we have 20 lines from autistic children, and they are matched controls we are working with. We can do with these things which were not possible with animals. We use this, for example, to show first already in May of 2020 that COVID-19 is infecting human brain cells, because we had the system and our colleagues had the virus, and it was easy to check. But we also showed, for example, in this example, a paper from 2021 that a risk gene for autism, SHAT8, and chlorpyrifos, a pesticide which is known to have some liabilities here, synergize in producing a phenotype which we would call autistic in our brain organoids. And this then led the NIH to create an Autism Excellence Center. We are now monitoring 175,000 children in 18 centers, and they form exposure gene hypothesis, and we are doing this then in our brain organoids to verify experimentally, is there really such a synergy? Something you cannot do with any animal. And this is perhaps the most science fiction type of research we're doing in this field, because we are combining our MPS via electrodes, microelectrode arrays, with an AI system. This little jumper here is actually controlled by a brain organoid. And it is learning to jump at the right moment to avoid these obstacles. We use this video environment to illustrate what we're doing here. These are only partially published results so far, but we have received about $2 million from Hopkins already to invest into this with our partners, and there's an increasing batch of articles around this system and how we're making progress in developing memory and cognition as a very simple form of cognition. You can imagine what this means for pharmacology and toxicology. We call it organoid intelligence because this is possibly giving us a system of memory and learning. Oops, I have to get back, sorry. A system of memory and learning, which we can do with patient backgrounds. We're doing this with Alzheimer patients, brain organoids and others, and we're looking into, do they learn differently? Do they get better? Do they memorize it until for the next day that they improved the game? I think this is showing you quite nicely, and it's bridging for me to AI. This is showing you how human skill levels have been achieved in different aspects of human capabilities. The baseline is the AI system is worse than any human, and the 100% line means the AI is outperforming any human being. And you can see to the very left of this picture here that the, it's working actually. This is human handwriting, deciphering human handwriting. The AI took 18 years to learn this. But on the very right, you see in orange, for example, coding. In only 2 years, AI learned to code, to program computers so well that last year, about 80% of all coding was done by machines, and all of the big tech companies have laid off coders. Yeah? In only 2 years, they outperformed humans. And it's only a matter of time for me until toxicology becomes also an AI-driven technology. And pharmacology actually was already a bit earlier, because since 2019, roughly, AI was good enough to design some drugs. And in 2023 already, 18 AI first drugs, so AI suggested drugs made into human trials, phase 1. And last year, the first one successfully passed a phase 2A and went into phase 2B with extended patient numbers from in silico medicine, a lung fibrosis medication. And imagine this, since 2019, that's when their project started, to only 2024 for preclinical development in such complex disease. This shows you the value proposition of these type of tools. 1 day earlier to the market is worth about $1 million. Yeah? That's what it is. So I've written extensively about the possibilities of AI in toxicology, how toxicology became a big data science with the high content, omics and imaging technologies, high throughput robotized testing, sensor technologies, literature and so on. And in the end, I think that we will soon have to write toxicology with AI. And the quick definition of this is, for me, toxicology is making big sense from big data with big computers. And that's what is at the moment entering. And alone, the data retrieval aspect of this is incredibly valuable. I was peer reviewer for the EPA of a risk assessment on a Superfund site chemical. 2 people had worked on this for 18 months to bring together all of the data on these data poor substances. 2 people, 18 months. After I did my peer review, for the fun of it, I used the AI systems and I found all of the sources in 1 hour. And I found even a complete risk assessment the EPA colleagues had missed, done by the fragrance industry. So you can see the value proposition of just getting the data and being more comprehensive. But then it can integrate pathology data, multimodal things, and help us with our reporting. To give you a bit of flavor, BioBricks is a project of ours where we have this paper, I think, is being hopefully accepted these days, is a way of importing entire databases with 1-line command. And we have written codes and shared already mostly on GitHub that with 1 line you can import an entire database without knowing how it is programmed, and you get in a standardized way all of the information. We have 140 of these. The first 12 of these databases gave us in total 117 million chemicals for which we have a chemical, a property, and a result. And 254 million of these triplets were generated on these 117 million data points. That's a treasure trove. 4,000 of these activities have more than 1,000 entries, which means for us it is good enough to cover the chemical universe. And these 4,000 properties you get in seconds, and the average accuracy is 91% in the training set. Compare this to 60 to 80% accuracy for 10 animal tests. So it is really about, at the moment, about building trust into these systems and making sense of this abundance of information. Suddenly knowing 4,000 things about a chemical is not making your life necessarily easy because you need to interpret these 4,000. This is a paper with Rob Califf, the former head of FDA, which came out just in April, where we're discussing how regulatory science at the moment has to live and stand up to this artificial intelligence. And this is also a series of articles about the aspects of validation, the challenge of these new methods to validate them. It is showing us how AI can help us, but also discusses how we can possibly validate AI systems with their enormous change. One article is discussing how biomarker qualification, something close to your hearts, is possibly helping us to make things more human relevant. And I call this altogether next generation validation for next generation risk assessment. Important discussion. Let me quickly close with the two aspects I addressed. Green toxicology, so benign by design. Alex Mertens and my team is leading this. She wrote the first textbook on green toxicology, is an assistant professor now. And only 4 weeks ago, we met in Stockholm for a Nobel Symposium with the fathers of green chemistry, Paul Anastas and John Warner, behind this. And we did the Stockholm Declaration on the Future of Chemistry. And this is all about using the same technology, as I said, front-loading them. And this is the same thing like what investigative toxicology is trying by prospectively risk anticipation of the targets and chemical classes which are being used. This is a workshop we did with the pharmaceutical industry. And I see that especially now with more than 50% of the drugs being human antibodies or human proteins, it is of highest importance that we get microphysiological systems and others into use in order to carry out this work. The last aspect for me is the big integration. I believe that ultimately we have to integrate genetics and exposure. This is a very easy line. The genes load the gun, the exposure pulls the trigger. And by mapping the human exposome similar to the human genome as a counterpart, we could put a safety on this. And with this idea, we carried out in May an exposome moonshot forum in Washington with a lot of European colleagues endorsed by WHO, UNESCO, the OECD. We had a lot of high-level speakers there. And in this meeting, I think we really set a starting point towards creating a human exposome project. Sven Asili, my deputy, is doing this. Again, AI is in the center of a lot of this because only AI can help us to understand the complex biomonitoring data and the patterns we see. We did sign a declaration towards a global consortium for the human exposome with the 300 participants we had, and I hope that this will be the starting point of something big and new. The last slide and statement here, I think that a lot of science are on green. You might have followed that in April this year, the FDA announced a roadmap to reducing animal testing, singling out monoclonal antibodies as their first target, but giving deadlines for moving out of animal testing. The very same day, Seldin, the new head of EPA, renewed their pledge to move out of animal testing by 2035, something already done by Andrew Wheeler in 2019. And 10 days later, the NIH came with this press release on to prioritize human-based research technologies, the microphysiological systems, to go hand in hand with this. Europe is at the same time talking about a roadmap already for a number of years. We are expecting a roadmap to phase out animal testing by early next year. So I put you on yellow, not yet, yet quite green, but we also saw already that the European research area which was just announced, has singled out this as one of the priorities for moving forward. With this, I hope I have shown you it is now or never. We have some disruptive technologies which we can integrate with existing knowledge by AI. I think this is a value proposition for accelerating product development and is going to optimize prevention and healthcare. I hope that the prospects of human exposome and a greener production, a production of substances which are safe and sustainable by design, as Europe is calling it, is a prospect which helped us all. Thank you very much, and I hope we have time for some questions.
Many thanks, Thomas, for a very inspiring talk here on, I don't know whether we are on amber when it comes to moving out of animal testing, but we are close to getting green. I don't think we have made so much noise about it as other parts of the world have done. I'm pretty sure we have a question here for you because some of our audience was attracted by your brain organoids, and when is an organoid suddenly being a real brain?
I think that cognition is a functionality of the brain. And the big thing about microphysiological systems is to recreate architecture and functionality. But the fact that these systems can react and learn something does not yet mean consciousness, sentience, self-awareness. We are very far from this. But we are working very closely with a number of bioethicists. There's an increasing interest on their side, even going into experimental ethics to see how much it takes to build a system. Our standard brain organoids are 500 micrometer, have 50,000 neurons, which is about as much as a housefly. And nobody will believe that they are being trained only on some very simple computer games could have any way of consciousness or self-awareness, but we have to assume that this can happen. Our largest systems we are building at the moment are 1 centimeter, and this is already 2 mouse brains, and they could very well, if we feed them with more information, at some point assume some type of consciousness. Yeah.
Interesting. Definitely. Other questions for Thomas? There is one coming up here, yeah? No, but these are linked to climate change by using...
Yes, the exposome is not meant to be just your chemical environment. It is also the social environment. It is the physical stressors. So there is subgroups. There's a lot of climate change exposome work. We also have discussions whether it's only about humans or whether they have an ecosystem, which is also to be integrated. It is clearly starting with the human exposome to match the human genome at the moment and also to integrate here, but we should be open for all of this. There's also increasing interest in microphysiological systems from animal species, from fishes and others to see whether we can create possibly information sources very similar to the human systems I'm working on.
Excellent. Many thanks, Thomas, and once again, thanks for coming and giving us presentation.